Module 4, Objective 4, explain the characteristics of skeletal muscle fibers and identify the structural components of a sarcomere. This particular objective is long and it contains a lot of terminology, so you're going to want to watch this several times and really take your time to learn the terminology. It becomes very important later in this module. So the skeletal muscle fiber is surrounded by a cell membrane, but we call that cell membrane the sarcolemma. In fact, sarco refers to muscle. So the sarcolemma is nothing more than the plasma membrane of the skeletal muscle fiber, and it surrounds sarcoplasm, which is the cytoplasm of a skeletal muscle fiber. We're going to find different organelles or variations of organelles found within these skeletal muscle fibers. So we have to sort of systematically go through so that we can understand the function of these organelles. And the next organelle is the transverse tubule. Transverse tubules are tubes that extend from the surface of the skeletal muscle fiber deep within the sarcoplasm. And their job is to transmit the electrical impulse or the action potential. All right, action potential is the electrical impulse that is going to be responsible for initiating the contraction of the skeletal muscle fiber. So it is transmitted or carried by the transverse tubules. Often along the sarcolemma, you're going to have openings that will then carry this message deep inside of the skeletal muscle fiber. And that is the role of the transverse tubule or the T-tubule. Here in yellow we see the transverse tubules that are running deep within the sarcoplasm. As the transverse tubules traverse deeply into the sarcoplasm, you're going to find another organelle called the Sarcoplasmic reticulum, or the SR. This is a network of tubes that surround a structure we're going to see in a minute called the myofibril. This organelle is very similar to that of the smooth endoplasmic reticulum, but they have these chambers that are located often very closely to the T-tubules, called the terminal cisternae. So the terminal cisternae of the sarcoplasmic reticulum are just little expanded pouches that occur near a transverse tubule. When you take one transverse tubule and two terminal cisternae of the sarcoplasmic reticulum, you get a structure referred to as a triad. And the triad is specialized for storing and releasing calcium ions. We had noted previously that one of the functions of calcium is to initiate muscle contraction, and this is where that calcium is stored prior to contraction. So these calcium ions are going to be actively transported or pumped from the cytosol into the sarcoplasmic reticulum, into these expanded pouches of the sarcoplasmic reticulum. So you physically have pumps that are active, so they require some ATP. We're going to convert ATP to ADP. And as that energy is utilized, calcium ions, it's really poor A, are then pumped into the sarcoplasmic reticulum. And that's where it's stored and that's where it's released to initiate muscle contraction. Myofibrils are subdivisions within the skeletal muscle fiber. Here we have a skeletal muscle fiber, and you can see here are the myofibrils, and there are going to be several in this image. These are the structures responsible for muscle contractions, and they're made up of protein bundles, or filaments, called myofilaments. We have two types of myofilaments. We have thin filaments, which are composed primarily of the protein actin, and thick filaments composed primarily of the protein myosin. So actin and myosin are the thin and thick filaments that make up the myofilaments that are found within the myofibrils. And as you can see just from that, there is a lot of terminology here that you have to come to terms with. So here we have just a cross-section of a skeletal muscle fiber. You can see the sarcolemma, which is the plasma membrane of the skeletal muscle fiber. And the sarcoplasm is everything inside. Inside, we have bundles of myofibrils. Bundles of myofibrils. We can see 1, 2, 3, 4, 5, 6, 7, 8, 9 on this side, and of course there's a whole bunch over here as well. But these myofibrils are just bundles of these thin filaments that are predominantly actin and these thick filaments which are predominantly myosin. Then we have surrounding each of these myofibrils networks of the sarcoplasmic reticulum here in blue. And in close conjunction with the sarcoplasmic reticulum, we have these T-tubules, which start out as openings on the surface of the sarcolemma, and they're going to then carry nervous signals down throughout. It's almost easier to see how these two T-tubules, they're going to surround each of these myofibrils. So they're very plentiful. And we're going to see how they function in a little bit. So lots of little parts here that you have to come to terms with. If you look at the muscle fiber, the smallest functional unit of the skeletal muscle fiber is called the sarcomere. And sarcomere is ultimately defined as the space between Z lines. And you really don't know what those Z lines are yet, but we'll cover them shortly. So interactions between the filaments within these sarcomeres, the thin filaments and the thick filaments, are going to produce contractions. And it's the arrangement of these thin and thick filaments which give rise to the striated pattern of the skeletal muscle fiber. Remember, skeletal muscle fibers looked striped or striated, and that's due to the arrangement of these thin and these thick filaments. When we look at the sarcomere, or the skeletal muscle fiber, from this perspective, from having this striated patterns, we start to identify bands, and we start to identify lines and zones, and that's what we're going to introduce next. So dark bands are referred to as the A bands, and we can see the A band right here. It is dark because this covers the thick filament of the sarcomere. So here in purple, we see all of the thick filaments. Remember, the thick filaments are the myosin proteins predominantly. The lighter bands are referred to as the I bands. And here we see the I bands, and the I bands predominantly have the actin protein. The M line is at the center of the A band. These are proteins used to help stabilize the positions of the myosin filaments. The H bands are on either side of the M line, and they have thick filaments but no thin filaments. And the zone of overlap is the very dark region where the thick and thin filaments overlap. We can see them on this image if we were to back up one. So the M line down here is at the center of the sarcomere. We have the zone of overlap where the thick and thin filaments overlap one another. And then the Z lines are located at the ends of the sarcomere. The I band contains thin filaments but no thick filaments. The Z lines bisect the I bands. They're the boundaries between the adjacent sarcomeres. And another molecule called titin, which has an elastic property or protein extends from the tips of the thick filaments to the Z lines, and they're going to help keep the filaments in alignment with one another, and they help restore the resting sarcomere length after a contractile state. Here we see that same image, perhaps a bit larger. And again, you're just going to have to commit this to memory and understand what constitutes the various bands and lines and why they're significant. And I cannot just lecture through that. It's going to take time, and it might help to sit down and actually draw this out. Here's an actual scanning electron microscopy or micrograph. Sorry, it's transmission electron micrograph, TEM, showing again the patterns of light and dark, where the A bands represent the dark patterns and the I bands represent the light, and that's what gives rise to that striated appearance. Muscle contraction is caused by the interactions between these two filaments, the actin and the myosin, and it's the structure of these proteins that determine the interactions. Thin filaments contain the protein F-actin for filamentous actin. In addition, there's other proteins like nebulin and tropomyosin and troponin, which play a role. Filamentous actin are twisted strands composed of two rows of globular proteins called G-actin. So basically the G-actins are all linked together, sort of like beads on a string. And if you get two rows of these G-actins, G for globular actin, and you twist it, you get your filamentous actin or your F-actin. Nebulin is a protein that holds the F-actin strands together. Other proteins involved with muscle contraction are tropomyosin. We're going to see that they're going to cover the active sites on those globular actin molecules. If you look at a globular actin, remember they're arranged like beads on a string. There is a region of this globular actin that is called the active site. And tropomyosin covers that active site when we're not contracting. Troponin is another protein. Troponin is a globular protein that binds calcium. Okay, so it's going to bind calcium. It's already connected to the tropomyosin and the G-actin, and we'll see its role shortly. Here's a better view. So if we were to take the sarcomere, right, the functional unit of the skeletal muscle fiber defined by this area between the two Z lines, and we were to blow up a little region of that, okay, we have that here, and then we were to even blow that up further, what we would find would be, again, the filamentous actin. Filamentous actin, the F-actin, are these long strands that resemble the beads on a string, and they're made up of globular actin, which are these little spheres, spherical proteins within the F-actin. And the globular actin has an active site, and if you look closely, there's a little yellow portion of each of these globular actins. That is the active site. It's identified here. And the active site during non-contracting phase is covered by a molecule called tropomyosin. And we can see this strand runs along the F-actin strand, and it's going to physically cover the active site, the myosin binding site. Lastly, we have troponin. Troponin is connected to the tropomyosin. It's connected to the filamentous actin or the F-actin, and it has a binding site for calcium. So to initiate contraction, calcium has to bind to its receptor on troponin, and that's going to cause the troponin-tropomyosin complex to change shape. ultimately exposing the active site on the F-actin. Now that we've covered actin, let's look at myosin. The thick filaments, which are predominantly myosin, contain hundreds of myosin molecules. Each myosin molecule contains a tail and a head. It often, to me, looks like a golf club. The tail is going to bind to other myosin molecules, as we'll see in a minute. And the head is made up of two globular protein subunits that projects towards the thin filaments. So there's two subunits here that sort of project towards the thin filaments. The core of the titan is going to recoil after stretching, and we'll see that shortly. All right, so here we have the two proteins together. Here we have a better appreciation of the myosin. So you have myosin tail, you have the myosin head, which is composed of two globular proteins, and you can see them, one there, two there, and a bunch, up to 300 of these myosin heads are all gathered together, found right here. And of course, titin is this green pigment protein that ultimately kind of holds it in place. So that makes up the thick filaments, and we see the thick filaments right here. And lo and behold, the thin filaments, the red fibers, are the actin. And you can see that the thick and the thin filaments, they overlap right here. So the myosin action. During contraction, the myosin heads, they interact with the actin filaments, forming a structure called a cross bridge, which we'll cover in just a second. The myosin additionally pivots, and you can see the pivoting sort of indicated by this arrow. It changes shape, and it produces a motion where the myosin head moves to a new formation. Again, lots of little details there, but if you spend the time to memorize them, life will be easier as we progress through this module.